Ophélie, juste pour répondre, en fait, on attend un peu, mais je pense que là, tu dois nous entendre.
Good morning, everyone. Welcome to this seminar in Ecology and Evolution of Montpellier. So a few technical details before starting for those of you attending remotely. You are in webinar mode, so this means that your mic and cameras are automatically switched off. If you have any technical issue, please send us a message in the chat and we'll try to help you to solve it. And if you have questions to ask to today's speaker after his talk, you can write them down in the Q&A tab, and we'll give you the floor when it comes to the questions to ask this question directly to the speaker. And without waiting more, I give the floor to Nicolas Rode, who will introduce today's speaker.
Thank you, Bruno. Hi, everyone. It's my pleasure to introduce Jackson Champer. Jackson did his PhD with Marcus Calcom at Beckman Research Institute in California on the west coast of the US. He worked on the development of antifungal vaccine. Then he did a postdoc with Philip Messer and Andy Clark at Peking University on the New York State in the east coast of the US. And he worked on gene drives, the topic of his talk today. And he works on different aspects of gene drives. So he worked both on molecular and population genetics. And also did experimental evolution in the lab, and also some computational modeling to predict and to better understand the dynamics of gene drive. And since last year, he started a faculty position at Peking University in China, where he's still working on gene drive and developing them in flies and mosquitoes. With this, now, Jackson, the floor is yours.
Hello, thanks for the introduction and inviting me to give the seminar today. I'm always very happy to talk about gene drive, which is still the main topic of my new lab here. So today we're going to cover 3 different projects involving gene drive. One experimental, one more computational, and one that's a mix, which is roughly representative of the different projects in my lab right now. So first of all, what is a gene drive? You've got a normal allele in yellow over here in a heterozygote. So this allele, on average, will be inherited by half of the offspring of this allele heterozygote. And that means if there are no fitness consequences of that allele, the expected allele frequency will remain constant over time in the population. A gene drive allele, though, is able to bias its inheritance. This allows it to expand in the population. This gene drive heterozygote, for example, where the red side is the gene drive allele, can potentially pass that gene drive allele on to all of its offspring. So that means very rapidly, even if we start with a low frequency in the population, the gene drive can increase until it takes over the entire population. So this is potentially a very powerful technique. If we develop a gene drive in a laboratory organism, we might just need to release a small handful into the wild to modify the entire wild population. There's 2 basic types of gene drives. There's the modification drive, which does exactly what it sounds. It's designed to change the target organism, usually by bringing in a cargo gene that has some kind of desired effect. such as preventing mosquitoes from transmitting the disease. But another popular type of gene drive is the suppression drive, which essentially is designed to reduce or eliminate the target population. We'll talk a little bit about how it does that later. So the main applications for gene drive that get most attention are, of course, mosquitoes. Right now, malaria will kill 500,000 people per year, mostly children, and other mosquito-borne viral diseases can cause hundreds of millions of cases per year. So Anopheles is the big one for malaria, and it's mostly Aedes mosquitoes as well as some Culex mosquitoes that spread the viral diseases. So together, these diseases are extremely important from a medical perspective, and a gene drive might well be able to stop them, even in areas where the mosquitoes are resistant to pesticides or where there's very low resources available for control programs, such as sub-Saharan Africa. But there's quite a lot more you can do with gene drive as well. If you've got a suppression drive, you can target invasive species such as carps in certain rivers, cane toads in Australia, kudzu in many areas of the world, rats in tropical islands. You can even use gene drive for economic purposes, such as targeting these invasive spotted wing Drosophila. With a gene drive, if you target a population, you can suppress it in an ethical manner. You don't have to worry about causing too much pain for animals. When dealing with crop pests, you don't have to worry about potentially hitting some valued insects when spraying pesticides. The gene drive will be extremely precise and targeted if it works. So there are also some scientific applications for gene drive. Overall, if we could get working gene drives, there's quite a lot we could do. So far, gene drive studies are in their infancy, but they have worked in yeast, a few different species of insects, mostly flies and mosquitoes, as well as mice. So there's really quite a lot of potential. The most well-studied form of gene drive is the homing drive, particularly the CRISPR homing drive. It's got a CRISPR-Cas9 allele together with some guide RNAs, and these cut the wild-type chromosome opposite the gene drive allele. If the chromosome has a cut, it's going to need to be repaired by the cell. Otherwise, the cell will be non-viable. But fortunately, cells are very good at repairing their DNA breaks. In this case, the cell is going to use homology-directed repair, lining up DNA on either side of the cut with DNA on the gene drive chromosome. So this means it lines up the DNA on either side of the chromosome because that DNA is identical to the DNA on either side of the cut. And then when it repairs the cut, it actually uses this gene drive chromosome as the template. And thus, it copies in the gene drive allele to this break. And now, instead of having a gene drive heterozygote, you've got a gene drive homozygote. Since this happens in the germline reproductive cells, that's how, even if an individual starts as a heterozygote, it can pass on the gene drive potentially to all of its offspring because its germline reproductive cells are homozygous. That's the theory anyway. Unfortunately, gene drives usually have problems when we actually try to build them in the laboratory. One of the most important and well-known problems is the problem of resistance alleles. If instead of homology-directed repair, we get end-joining repair, then the gene drive allele will not be used as the template for repair of the wild-type chromosome. Instead, these two ends will just get stitched back together, and during this molecular process, the sequence is changed. Now that this sequence is changed, it's no longer a match for the gene drive's guide RNA sequence. And that means the gene drive can never cut this allele. That's why we call it a resistance allele, because it's resistant to cleavage and thus resistant to this process of drive conversion. Even if a gene drive is very successful, you can see the red allele frequency here, increasing to 99% in just a few generations. If we make even a small number of resistance alleles during that time, and these resistance alleles have a fitness advantage, which they almost certainly will, well, then the resistance alleles can quickly take over from the population and any positive effects of the gene drive would be lost. In this case, they can do it quite rapidly in just a few dozen generations. So how can we potentially get around that problem? Well, the first thing is to think about different types of resistance alleles. When we get end-joining repair, we can get all sorts of different insertion and deletion combinations. And most of the time when we're doing gene drive, we are targeting a particular gene of interest. This means that when we repair that gene, we can get either a functional repair or a disrupted sequence. The disrupted sequence will usually happen if there's a frame shift, for example. Then all the rest of the amino acids in the target gene will be completely different. But even if there's not a frame shift, much more often than not, we're still changing enough of the amino acids to prevent the target gene from functioning. This kind of resistance allele is a little bit less serious. We can potentially come up with some ways of dealing with it. But if we get a functional resistance allele sequence, that's the kind that's going to have the really big fitness advantage over the gene drive. That's the kind that's going to be able to take over from the gene drive in the population. So how can we avoid that? One promising solution that has been well tested in our lab is using multiple guide RNAs. Instead of just one guide RNA target, we cut several adjacent spots in this target gene. In this way, they would need to all be repaired perfectly to get a functional sequence. If even one of these is repaired imperfectly, like in this example, then the whole target gene will still be disrupted. And of course, if we, instead of cutting these sequentially, if we cut out a big piece by cutting at several of these in the same time, that's also going to provide a big enough disruption to the target gene to render it non-functional. So by using multiple guide RNAs, we can make sure that we don't get these functional resistance allele sequences. At first, it was also thought that these multiple guide RNAs would increase the frequency of drive conversion. But in reality, all this cutting happens at about the same time, and we only get very limited increases in the drive conversion rate, the rate that wild-type alleles are converted to drive alleles when we use multiple gRNAs. It's best at just preventing this functional resistance. So we wanted to actually test all of these together. We made a gene drive that targeted RPL35A, which is a haplolethal target gene. That means that all organisms need 2 working copies of this gene to survive. If they only have 1 copy, then they're going to be in trouble. We used 2 guide RNAs in this example to make sure all resistance alleles would make this target gene non-functional. And then there was one final important piece of the gene drive, and that was the recoded version. We were trying to make a modification drive over here, and it's very important that the gene drive itself stays viable. So the gene drive, because it has this recoded version with a different sequence than the guide RNAs, will always have a working copy of this target gene. Unlike complete gene drives, we didn't include the Cas9 as part of this gene drive. We provided this at a different genomic source. This way, if these flies got out into the wild, they wouldn't necessarily spread the gene drive to the whole wild population. This split element would make sure that it was safe to study this gene drive in the laboratory environment. So what was the idea behind targeting this important gene? In the germline, we ideally convert this blue wild-type chromosome into the red drive chromosome. So this is what we want to happen. But sometimes we get a resistance allele at this wild-type chromosome, converting it into one of these yellow resistance alleles. And then meiosis takes place, and then fertilization. And then we've got these 3 genotypes. These ones are fine since they have 2 working copies of the target gene, because the gene drive has that recoded sequence. This one, though, is not viable. It has this resistance allele, that has a broken copy of the target gene. And even though it has one functional wild type copy, this gene is so important that you need 2 copies. So these guys are then immediately removed from the population. And that's good. That means we get rid of the resistance alleles. Unfortunately, gene drives often deposit a lot of Cas9 and guide RNA into these early embryos if it was the mom that was a gene drive parent. So this gene drive mother can now keep cleaving all of these wild type alleles in the embryo. And that means that some of these embryos will be rendered non-viable if we get this additional resistance allele formation. But fortunately, in this gene drive, this only happened some of the time. So we were still able to get lots of drive offspring that survived and could then keep on spreading the gene drive. So the idea was we get rid of the resistance alleles, leaving only the gene drive alleles, which can then keep spreading through the population. When we released these into the population, and this was a little bit of a high release, higher than we intended, but sometimes there was some uncertainty in these cage experiments. For this gene drive, though, that wouldn't have mattered. Whether it was high or low, it increased very rapidly in the population until it took over the entire population. We did a computational analysis of this frequency trajectory, and we found that this gene drive didn't seem to have any major fitness costs. Their 95% interval was actually centered around 1. So this was the first successful homing modification gene drive. We're also quite interested in suppression gene drives because other labs, especially the Casanti lab at Imperial College London, has had a lot of success in bringing suppression gene drives to Anopheles malaria mosquitoes. In Anopheles, the drive conversion rates are very high, much higher than in the fruit flies that we were doing experiments in. But we were mostly interested in seeing if our multiple gRNA method can still work well with a suppression gene drive. So how do these suppression gene drives work? Instead of targeting a haplolethal essential gene where 2 copies are required, a suppression gene drive targets a gene where only 1 copy is required. So that means these drive wild type heterozygotes are completely fine. The other aspect of a suppression drive is that the drive doesn't have a recoded sequence. So this drive is actually breaking the target gene. This individual only has 1 good wild type copy, but it's still okay because this gene is haplosufficient. 1 copy is enough to be healthy. And when making suppression drives, we prefer to target female fertility specific genes so that males can still be fine. We're only affecting female reproduction. Because these heterozygotes are still fully viable, we can have a lot of heterozygotes in the population, which increase the frequency of the gene drive. Eventually, we get more and more homozygotes in the population, and these homozygotes will be female sterile. So that's how gene drive can spread in the population at first, and then when it gets to high frequency, it starts suppressing the population. This worked quite well in mosquito tests, suppressing laboratory populations, but that was with just 1 gRNA. In this test, we did it with 4 gRNAs. And as you can see from these tests, the gene drive frequency stopped at around 60% or so. So that's not good in that we failed to suppress the population, but if we had generated resistance alleles, then the gene drive frequency would have started going down. So it was a partial success. We were able to use multiple guide RNAs to avoid resistance alleles, but because we didn't have the high drive efficiency seen in mosquitoes, we didn't have enough power to suppress this very robust fruit fly cage population of a few thousand. All right. So that was some of our fly experiments using multiple gRNAs. How would these gene drives actually work in more realistic wild populations? After all, a lab population is simplified in many ways. It's a cage, so any individual could find any other individual pretty easily to mate. And that's how a lot of computational modeling is done as well. Sometimes computational modelers will have these 2 separate populations with migration between them. But in many cases, it would be much more realistic to model a landscape where individuals can move freely in 2 dimensions and only mate with their neighbors. You can even get very complicated landscapes with rivers, swamps, agriculture areas, urban areas. All of this could affect how a gene drive spreads. But skipping straight to a super complicated environment like this is a bit too much to start with. Instead, we just had a 2-dimensional environment where all the space is the same, but individuals could still be scattered around this environment and only mate with individuals near each other and only have offspring with individuals near each other. So in this situation, we're going to release a bunch of suppression gene drive individuals in red, and then each frame is going to be a different generation. You're going to see these gene drive individuals spread throughout this population. This particular movie might be a little bit jerky, but we've got a workaround for that in a minute. So in this one, you saw a very successful population suppression gene drive. It took a little bit longer to suppress the population than in a cage experiment, 23 generations compared to maybe 10, because the gene drive had to migrate out to the edges of the environment. But that's not super interesting. Things get more interesting when we look at gene drives that have slightly imperfect performance or environments when the migration is lower. So we've set up a YouTube movie that will allow you to see a smoother version of that movie, so you can see exactly what is happening. Should I stop sharing my screen now so we can put on the YouTube movie?
Yes,
Oh.
we can do that.
Right. Yeah, my China internet connection has a little bit too much lag to show the movies, even though everything else is okay. So in this movie, you're going to see a phenomenon that we call chasing. So this is where the gene drive starts in the middle, and at first it expands outward, suppressing the population. But some wild type individuals then are going to manage to sneak through the expanding ring of gene drive and move into that empty area in the middle where the gene drive has suppressed the population. When they're in the empty area, there's going to be much less competition between individuals. And so those wild type areas that escape to the middle are going to have many offspring, and they're going to do very well. And the gene drive is still going to be present, chasing after the wild type, but you'll see it never quite manages to catch up. So you can see at the bottom there, some wild type escaped, and now those wild type are going to move into the empty areas and have a whole bunch of offspring. So even though the gene drive is still there, you're going to see that the gene drive never quite manages to catch up in that situation. So these periods of chasing can actually last quite a long time, sometimes indefinitely. We've often done tests, and chasing can last, in most cases, for over 1,000 generations. Sometimes they'll only last for a few dozen or a few hundred generations before we get suppression, but oftentimes it simply never ends. Now, you might think, okay, well, clearly we reduced the population compared to the normal situation. I'll share my presentation again. So clearly we reduced the population compared to the normal situation. But what happens then if instead of a completely perfect gene drive, you're a little bit imperfect? Well, in this case, you could still have chasing, and you can still sort of see it here, even though it's more jerky than before. But the longer chasing happens, the bigger the chance is that we form a functional resistance allele. That's yellow on the left side over here. And now you can see that that functional resistance allele is very quickly expanding and taking over the whole population. Suppression gene drives are even more vulnerable to functional resistance alleles than modification gene drives. As soon as one of those forms, it's game over for the suppression gene drive. That's why we were so interested in using multiple gRNAs in a suppression gene drive. The Anopheles mosquito gene drive that had just one gRNA is a great gene drive, but there's a difference between suppressing a population of several hundred and suppressing populations of tens of millions in the wild. We wanted to develop these multiple gRNA techniques as kind of some extra insurance to make sure that this resistance never comes into play for a suppression drive. So in addition to our discrete generation models, sort of simplified models of gene drive spread, we've actually modeled mosquitoes in particular. You can see a few different outcomes in these bars based on the type of drive. These red outcomes are the kind that we want to see, where we get nice rapid suppression without any chasing phenomenon. Purple is when we do get chasing, but it's for a limited time. Sometimes for a very long time, but at least not forever. At least we get suppression, after maybe 20 years or so. And then this yellow color is where we get chasing without any suppression for over 20 years. You can see in the discrete generation model that some of these gene drives that people are thinking about in mosquitoes can actually do fairly well. But when we used a mosquito model, we got almost entirely chasing except for the very best gene drives. And these best gene drives, unfortunately, this one over here, NOS-F3, is very much a hypothetical gene drive. And NOS-F2 is probably the best we can possibly expect to get in a real laboratory environment, at least in the near future. The most likely gene drives that we have available now are NOS-F, ZPG2X, and ZPG2. And as you can see, these were 100% chasing in our model. Compared to our discrete generation model, mosquitoes just have tons and tons of offspring, and it's only the offspring that compete with each other. The female mosquitoes, meanwhile, can just have so many offspring that even if we suppress the population a lot and there's not that many females, the ones that are there can still lay tons of eggs and have lots of surviving offspring. So suppressing mosquitoes in these continuous spaced environments is super, super difficult, even with the latest gene drives, and even if we're assuming that they don't make any functional resistance alleles. Or at least that's what we thought until some more advanced recent modeling we did that involved competing species. After all, in any realistic ecological environment, there's never going to be one species that just has the entire niche by itself. Or I shouldn't say never. In most of the time, a species isn't going to have its own niche that has absolutely no overlap with anyone else. Many mosquitoes, for example, might be human specialists, or their larvae are specialists to a particular environment, but there are still other mosquitoes that can bite humans. There are still other organisms that can use those resources that the mosquito larvae are utilizing, at least in some situations. So to better model that, we used not one type of species, but two different types of species in SLIM. And when it comes to competition, two members of the same species will have strong competition, but the other species will only compete a little bit with the target species. And the exact amount is dependent on a variable parameter in our model. The other thing we have in our model is seasonality. Right now, a lot of people have estimated that if mosquitoes are at low densities, they can grow very quickly. And that's informed a lot of gene drive mosquito modeling for both my laboratory, as well as many other good laboratories doing mosquito modeling studies in gene drive and for other reasons. But that doesn't necessarily mean that this rapid growth rate could apply all the time during the year. At the very beginning of the wet season, probably everyone is enjoying many resources due to low populations. But if we suppressed the population of mosquitoes in the middle of the wet season, well, then it might not be able to grow back quite as quickly as at the beginning of the wet season. because there's now a lot of these competing species around that also grew together with them at the beginning of the wet season, but are now still at their high frequency because we weren't suppressing them with the gene drive. So here's our data from that. You can see that these are the main outcomes based on seasonality, basically how extreme the seasons are. If we have very high seasonality, then the population is much higher during the wet season and much lower during the dry season. The competition factor is how much do the two species compete. Competition factor of 1 is pretty unrealistic because that means the species are 100% competing. That's ecologically unstable. But 0% competition probably also is also unrealistic because then the species are not even interacting at all. So you can see over here that long-term chasing that we're trying to avoid completely disappears with a moderate level of competition. And this is even when we're modeling a not so efficient gene drive. So in this case, the competing species, no matter what level of seasonality we had, was able to avoid this long-term chasing phenomenon. And we often got some suppression, either before chasing or after chasing, depending on how much seasonality. So in this more realistic environment, we potentially have much more successful outcomes, even with a gene drive that doesn't have extremely high efficiency. But there's still one problem. In addition to getting more successful outcomes, we got a different type of problem. Instead of long-term chasing, we had drive loss. So that's when the gene drive was able to suppress a chunk of the population, but then the gene drive disappeared before it can reach all the members of the population. So it seems like this competing species can sometimes create pockets of space where the gene drive eliminates its local pocket, but then some wild type manages to escape to other areas and then can eventually bounce back. Now, you could potentially just keep releasing the gene drive until it works, but it does potentially mean that we can't just release the gene drives once in one spot and expect them to be fully successful everywhere. We might need to take a little bit more care to release the gene drive over a wider area, or possibly do an occasional re-release of the gene drive in order to ensure that eventually we get one of these suppression outcomes. Of course, we'll need to measure how much these individuals, how much these target species compete with other local species in the area to know how practical this might be. Another important thing, of course, is to make sure that the competing species doesn't also have the capacity to transmit disease. It might be that a competing species that previously was mostly excluded from human areas does actually become a more important disease vector if it's able to replace one of these more important disease species that we target with a suppression drive. So suppression drives, could well still be a promising outcome, but in some cases, a modification drive might be a more reliable outcome if you manage to have a good cargo gene that's resistant to mutation. All right. So far, we've been talking all about homing gene drives. Homing gene drives are one of the most powerful types of gene drives, but sometimes that power can be a problem. This little diagram shows shipping routes of the world. Bottom line is that the world is very interconnected. If you have some species in one spot of the world, eventually it might be able to spread everywhere else that that species exists. And that means that a homing gene drive is not going to be a confined gene drive. You can, except for some perhaps very rare situations of extreme isolation, if you release a homing gene drive, it's probably going to spread everywhere in the world that that species is located. In some cases, that might not be acceptable. There could be all sorts of socio-political considerations or governmental regulation that might prevent use of an unconfined gene drive in those situations. And of course, when you're targeting an invasive species, you don't want it to spread everywhere in the first place. Maybe you would be okay with spreading an anti-disease gene drive everywhere in the world. if you could get different countries to agree. But if you're targeting an invasive species, you don't want that to spread back to where the species' native environment is located. Then you might be causing as big an ecological problem as you're solving by getting rid of the invasive species. You need a confined gene drive to do that. And in the lab, we do quite a lot of development of confined toxin antidote type gene drives. These all work via a simple principle. The drive allele isn't designed to do drive conversion, homology-directed repair. It does not cut the wild type allele and then copy itself into the wild type. No. The only thing that the drive does with Cas9 is cut the wild type allele and turn it into a disruptive allele, which is kind of the same thing as a non-functional resistance allele. The drive also has a recoded copy of the target gene, just like the haplolethal homing modification drive that we saw earlier. So even though the drive is cutting up the wild type allele and making a lot of these disrupted alleles in important genes, the idea is that the drive is still okay. So the TARE drive, toxin antidote recessive embryo drive, is the simplest example of this kind of CRISPR toxin antidote gene drive. In this case, we're targeting a haplosufficient but essential gene. So everyone is okay, both males and females, if you have one copy of the target gene. But if you don't have any copies of the target gene, then it's non-viable. So first in the germline, all we do is cut this wild type allele. There's no copying of the drive allele. And then we get meiosis and fertilization. So in this case, each of these two alleles split up, and now they're each together with the wild type allele. And now we're assuming that this parent is a female parent. So we get that maternal Cas9 and gRNA deposition into the embryo. This can now cut these wild type alleles, even in the embryos that don't have a gene drive, because all of the Cas9 was expressed earlier at this stage when the cell did have a drive allele. There's still a lot of Cas9 here that's maternally deposited, even though there's no drive allele. And then what's our situation here? This egg will be just fine. It doesn't have any wild type alleles, but it does have a gene drive allele, which has a recoded rescue copy that cannot be cleaved by the drive's own gRNAs. So that is a working copy of the target gene. This one, though, doesn't have any functional copies of the target gene. So this individual will be dead. So this means we have a frequency-dependent gene drive. The more gene drive we have, the more wild type alleles we can cut. So instead of working to increase the number of drive alleles while decreasing the number of wild type alleles like a homing drive does, this gene drive, it doesn't copy itself. It just gets rid of wild type alleles, thus relatively increasing in frequency. It's not quite as fast as a homing drive, but it can still get the job done. Here's some modeling that shows its performance. We're showing here the fitness cost of the gene drive, well, the fitness of the gene drive. So 1 is a perfect gene drive. Something like 0.8 or 0.9 might be more typical of gene drives that have a cargo that makes the insect a little bit less healthy. And this is the introduction frequency, how many gene drives we introduce into the population. You can see that if the gene drive has even a small fitness cost, then we need to be above this dashed black line for the gene drive to even theoretically be able to succeed in the population. Once we get into this blue area, then we've released enough individuals that the gene drive will then be able to spread to the whole population pretty quickly. If we stay in this gray area, then the gene drive will actually decrease in frequency and be eliminated from the population. So this is one thing that can make this gene drive confined. We need to have enough gene drive, otherwise it will disappear. So if just a few individuals appear in a port city, they probably won't be able to establish in that population because they're only a small fraction of the population. They'll probably be eliminated very quickly. So another way to think of that is using 2 different models. So here we've got a different version of the TARE drive that is a little bit more confined. We release them into an introduction deme, and then that introduction deme has some migration with a linked deme. you can see with this gene drive, we have to release quite a bit of it to avoid drive failure. But then if we release about 20% or more, then the drive will succeed. If the gene drive migration rate, or if the natural migration rate between these populations is low enough, less than 2%, then we'll be in this black area, which means that not enough gene drive individuals can arrive in the long term for the gene drive to invade this linked population. On the other hand, if the migration rate is high enough, then the gene drive can invade this population. So it all depends on the balance between the gene drive threshold and the migration level, if the gene drive is going to move from one population to another. So this means that any kind of gene drive with an introduction threshold can be confined. Exactly how confined? Well, that depends on the introduction threshold. We have a very large number of different gene drive designs, each with different gene drive thresholds. So hopefully this means that we have a gene drive for every potential confinement situation. All right, so tear drive, very promising as a combined gene drive. It's also very simple to build. You notice that that embryo resistance actually gave us even more of the disrupted alleles that we wanted to have with the tear drive. So the promoter for the tear drive is much more flexible than for a homing gene drive. So that means it might be quite a bit easier to make tear gene drive in non-model organisms as well. So not only is it confined, but it's also easier to make. We were quite interested in building this in our fruit fly model. So we built something that targeted the hairy gene, H. This is essential but haplosufficient. So if you have one copy, which can come from the recoded region of the drive or a wild type copy, then the individual will be viable. We use 2 gRNAs, again, to prevent the formation of functional resistance alleles. It's very easy to use multiple gRNAs for tear drives, even easier than homing drives. So these show some cage experiments. The blue gene drive here is the tear gene drive. You can see that we started at a little above 20%, and it increased pretty quickly in the population until it took over the population. And that was also the case in this other cage. So tear gene drive was definitely a very powerful gene drive that did require a high release amount for it to be fast, but this release amount is still pretty small compared to other genetic control techniques. And once the tear drive gets going, then it can easily go the rest of the way after just one release. In this case, we were interested in not just the tear drive by itself, though tear drive would make an excellent modification drive if used alone. We were interested in turning it into a tethered gene drive. So we do potentially have some options for confined suppression, but that's something that is a little harder to do with just a confined gene drive. The tethered gene drive idea, though, is all about using a confined gene drive to power a homing gene drive, especially a homing suppression gene drive. The idea is that the homing gene drive doesn't have Cas9 on its own. It's like a split gene drive. The confined gene drive provides the copy of Cas9. So the confined gene drive will only spread in the target population. And then you release the homing gene drive, and then the homing gene drive will only suppress the target population. It won't be able to move to other populations because the confined gene drive can't go to those other populations. And the homing drive needs the Cas9 in the confined gene drive to spread. So this was an example with our modification gene drive. We released the haplolethal homing gene drive, this time at a very low frequency, and it shot up to 100%. We did the same thing with the homing suppression gene drive, but it did the same thing that it did before when it was by itself, where it kind of got stuck at around 50%. But it still shows that the tear drive can work quite well providing the Cas9 for these, even though we'll need to increase the efficiency of our homing suppression drive for it to succeed in fruit flies. So there's quite a bit of other research going on in my lab. We've got lots of different kinds of mosquitoes and all sorts of ideas for how to make high efficiency mosquito gene drives. we're always working on more realistic computational modeling, not just spatial, but even more realistic life cycles, other organisms, geography and terrain. And we're always interested in starting collaborations for gene drives in other organisms of interest, whether those are other insects or even some model vertebrates. So I'd like to especially thank all of my lab members. We go on all sorts of fun adventures, eating delicious noodles and pizza. It's been a very fun, productive lab. They've done probably about half of the work that I showed you earlier today, and the other half comes from my previous postdoc. Feel free to check out our lab website for our latest papers and news for future papers and other gene drive stuff. So thanks very much. I'm happy to take questions now.
Thank you very much, Jackson. Are there any questions either in the room, but we are not that very much in the room, or online? There are more people. Nicola, please do not hesitate to moderate also if you want.
Yeah, sure. Maybe I can start and ask a question. If
Sure.
So I had a question about you showed at the beginning of your talk suppression drive where the drive got stuck at a frequency of around 60%, and you said that the reason for that was the efficiency of the drive. And I was wondering about fitness. So it's not I think it's this one. And I was wondering about fitness cost, because you expect this kind of coexistence between the drive allele and the wild type allele if there is a large fitness cost. So I was wondering if homing efficiency was the reason, or if whether it was that there is a really high fitness cost.
So fitness cost is definitely important, though in a complicated way. I sort of gave a simplified version of this. So when it comes to suppression gene drives, they have a certain suppressive power, which we often describe by their genetic load, how good they are at getting to a high equilibrium frequency and making a certain fraction of the females sterile. And if they make enough females sterile, that results in complete suppression. If not, the gene drive just reaches an equilibrium frequency. And that means that the total population size might be reduced, but not eliminated. The question is, what determines this equilibrium frequency, which determines the genetic load? The most important factor for that is the drive conversion rate. Just the rate at which the rate at which wild type alleles are converted to drive alleles in the germline of males and female drive heterozygotes. If that drive efficiency is super high, like in the Anopheles mosquitoes, then even if you do have fitness costs, formation of non-functional resistance alleles, you're still going to have a very high genetic load. If your drive conversion efficiency is lower, that by itself will reduce your genetic load, but it also makes things like fitness and embryo resistance allele formation rate very important for genetic load. So once you lose the drive conversion power, then lower drive conversion, fitness costs, and non-functional resistance alleles can all combine to make your genetic load even lower. In this specific scenario, we did indeed have all three of those things. The drive conversion was less than Anopheles, and that made it so that the high embryo resistance allele formation rates also reduced the genetic load. And when we did our maximum likelihood analysis, we detected some additional fitness costs, because even with those two factors, we were expecting to see it get to an equilibrium up here at 80 or 90% instead of 60%. The fitness costs in this case are a little mysterious, because we use the nanos promoter, which is excellent in Drosophila for being germline restricted. So we suspect that the target gene might be a little more complicated than just essential but haplosufficient female fertility gene. We've got another project that's trying to start looking at that in more detail right now. Long story short, it might, we might need a gene that's not required in the germline. Because remember, somatic cells look like this, but germline cells look like this. So we need it to be haplosufficient for somatic, and we need it to be not required in the germline, at least in the late germline. Though again, that's still, to a certain extent, speculation rather than things we've confirmed with additional experiments right now.
Okay, thank you. Are there other questions? I mean, I have another question, but I don't want to. Michael, yeah.
Hi, so thanks for the very nice talk. It was really clear. So I posted my question also in the chat. So I'm studying a lot the Gambier complex and all the interaction with the different species. So right now, the model that you are showing us, they are mainly designed for isolated species, or so what would be the consequence of thinking that one species like the Anopheles gambier or Ecologie is actually connected to a network of multiple species where they can produce fertile females? So how does your model, does that change anything in your model, and how would that change things?
So that's an interesting question, and the exact answer depends on exactly what we're trying to do with these species. I would say in most cases, you've got lots of malaria mosquitoes in this complex. So, you might not want to target just, the most well-known Anopheles gambii, but also the other Anopheles gambii in the complex and the other closely related mosquitoes. If you've got a homing suppression gene drive, or especially, if you've got a homing modification gene drive, let's say, this gene drive is going to spread everywhere. It can spread from very low rates. The moment it hops to another species, it's now going to be able to start spreading rapidly in that species. Which could very well be a good thing, because you might need to get into these other species to prevent malaria. For a suppression gene drive, things might be a little more complex. It all depends then on your exact rates. If there's a high rate of genetic movement between these species, it might be that your suppression gene drive would always jump from one species to another. And thus you would then suppress all of these malarial species. But if the speed of the gene drive is faster than the speed that it can jump, then that wave front of advancing gene drive might simply not have time to jump to the other species before it suppresses the target species. And then you might be in that competing species situation where another species takes over from the original species quite quickly, because all these species are definitely close enough that they would have high competition factors.
Right. And
in a situation like this, sorry, what were you saying?
No, go ahead. Go ahead, sorry.
Yeah, so in a situation like this, maybe ecological measurements could let you know if you'll jump to the other species, or if you have to do some other stuff to handle the other species, like put them together in the cage to get your gene drive into the other species, or maybe even make a custom gene drive in this new species if reproduction is extremely limited between them, in order to suppress all of them. The other thing to worry is that if there are any somewhat close members of this complex that are thought to not cause that much malaria, I'm personally a little bit worried that if they were able to explode taking over from the other species, if they dodge the suppression drive, then maybe they would become more important with malaria. That might be another experiment that could be important when thinking about the deployment of suppression gene drives. On the flip side, you've got confined gene drives, which again may be necessary. I'm thinking about them mostly for invasive species or agricultural pests, but again, there's still the socio-political considerations for even the very widespread mosquitoes. And if you have a confined gene drive, you might be able to get that going in your target species. But if you have only very limited hybridization between the two species, well, that might be a situation that is almost more akin to this 2D model, with this migration rate kind of being your hybridization rate.
Yeah.
And then you would need to have enough hybridization to avoid confinement of the gene drive to just your target, just the original species. The gene drive might actually not be able to hop to the other species, even if they have a fair amount of reproduction, if that hybridization level is still quite low compared to how much they mate with their own main part of the species.
Thanks. I do have a follow-up question, but I will leave the floor for.
Are there other questions? Maybe you could ask your second question, Michael.
Yeah, the second question was really following up on, do you also expect more resistance? Because indeed, currently the guided RNA that, well, the target site are mostly designed based on the most important vectors, without taking into account the reservoir of potential diversity that there are in those related species where they can form hybrids and brings resistant allele in a pool that were not, modeled beforehand. So would that, so even if you target multiple sites, so I guess that's currently not considered in these models.
Right. So far our models of multiple guide RNAs have always been super simple, just trying to understand more the genetics of it. And yes, of course, we see the benefit in the population as a whole, but we certainly haven't been explicitly modeling genetic diversity between different species, accessing all that genomic data. I do imagine that could well be an important consideration, even if we had an ideal target set in one species. It might not be quite enough to deal with another species. And this is a little bit more of a problem than you think. I didn't show these slides here, but as you increase the number of guide RNAs in homing drives, not in toxin antidote drives, but in homing drives, your drive conversion efficiency will be lower. At first, it will go up. 2 guide RNAs should almost always be better than 1. And 3, probably also an improvement, depending on the exact parameters. But after that, you're going to start losing drive conversion efficiency. And if you've got a suppression drive, well, you saw what happens if you don't have enough drive conversion efficiency. It can't even suppress the population in the first place. So you could try to overcome this problem by more guide RNAs, and maybe you could do it if you had that information available. But maybe the information wouldn't be available, and maybe it would require too many guide RNAs that you would lose too much efficiency. And in that case, maybe you would need to design custom gene drives for each one of these species complexes. The hope is that targets like double sex are super, super conserved, and you can deal with just a few guide RNAs. But I think some things about that are still a little unclear, especially if you talk about the possibilities for different resistance allele mutations in the different species.
Right. Thanks.
Are there any other questions? So maybe a quick last question, if I can. It would be about natural conditions, because I think that's really the difficulty, especially when we're talking about fitness and fitness costs. Maybe I'm a bit too focused on fitness, but all these things about this invasion threshold, for example, they really depend on the fitness cost, and this could vary in a realistic ecological condition. And so, how do you think? How should we take this into account when for researchers like you that are developing gene drives, this this ecological realism?
I think it's definitely very important, though exactly how important to a certain extent depends on exactly what you're trying to do. If you're trying to say use a homing modification drive, then yes, it is important to consider many things, but that's a simpler type of drive in some ways. things get really complicated if you're trying to do suppression or confinement. We already know, we've tested in genetically diverse Drosophila. Gene drive performance does differ based on genetic background, sometimes even in very important ways. So if we want to, if there's something we're particularly trying to avoid, and we want to make a model that can be, that will let us be absolutely certain we can avoid some undesired outcomes, such as invasion in a particular area that we want to protect, we will need a much more sophisticated model that can take these things into account. And even then, we're going to want to leave enough of a buffer region in drive performance that even if there's some factors coming into play that we don't expect, the gene drive will be confined. So even if our best models say, a gene drive with a 25% threshold will always be confined and not invade the population we don't want, well, we're not going to make a 25% threshold gene drive then. We would probably want to make at least maybe a 28 or 30% threshold gene drive then to have enough of that safety buffer so that even if our models are not perfect, we still won't get an undesired outcome. And that's, a model where we're already doing as much as we can to make it accurate.
Okay, thank you. I guess, Jeanne and Bruno, I guess if there are no other questions, thank you everyone for attending, and thank you, Jackson, for the nice talk.
Yeah, thanks again for the invitation.
Thanks a lot, Jackson. Bye-bye.